All right, so chapter 38 is about angiosperm reproduction, and the main idea here is that flowering plants are influenced by both the biotic and the abiotic environment. So we're going to talk about some of the things that are involved in causing plants to flower. Okay, so flowers, these are the reproductive shoots of the angiosperm or the flowering plants, the angiosperm sporophyte. So the floral organs are the sepals, the petals, the stamens, and the carpels. So these are the structures that really make up the flower, and you're going to want to be able to draw one of those and figure out what the different parts are and what they do and what they contain. So the flowering involves the change in gene expression at the apex of the shoot. So instead of them producing leaves like they would normally at the apex in that meristematic tissue, they're going to produce some flowers. And the flowers are, of course, the sexual structures of the plants. And some of the things that influence flowering are temperature, the length of the day, the amount of light that they're receiving, different types of things, and we're going to talk about these. But these are all kind of activators of the genes that are controlling the flowering. And specifically, it's the dark period that's going to be the main trigger for the flowering production. And some of these experiments from the 1940s that the USDA did were looking at the length of the day and the night and how those things were involved in really getting the plants to flower or to not flower. And those are referred to as the photoperiod. And what they found were short day plants, which are also called long night plants. These flower when the days are 16 hours or shorter. In other words, the nights, the dark period, those are 8 hours or longer. And what they were looking at, or looking for rather, is flowering. And they found that if the daytime period of the photoperiod is broken by a brief period of darkness, there's no effect. So in other words, if you're looking at the, oops, I'm trying to find my little tablet here. If you're looking at the daytime period right here, whoops, sorry, I got the wrong thing, got to get the pen going. So again, if you look at the daytime period here, if you break this up by a period of darkness, there really was no effect. And what they were doing was, is if they were interrupting this nighttime period now, if you look up here, if you see the nighttime period, if they broke this up with a little flash of light, they could find that they can get the plants to not flower. And then they, kind of discovered, as you see here, there's two different types of plants. You've got short day plants and you've got long day plants, and we'll talk about those as we go on. So if you look at the long day plants now, this is the case where when the long day are grown in a period of a long night, you don't get any flowering occurring. So if we look right here, these are long days, but they're actually responding to this long night. And again, we get no flowering here, okay? And then here, this long night, short day. Now if we get a long day and a short night rather, so they're called long day plants or short night plants, this short night, this is going to respond in a flowering, as you can see there. So what they were doing is they were just playing again with the photoperiod. And we'll get into some more of the details with this in another chapter as well, okay? But what they found in these experiments was that red light was most effective at interrupting the photoperiod, or the nighttime portion of it. And it was because of this phytochrome protein that you see here, just kind of depicted. They almost looks like some rabbit ears. And what they're doing is measuring the photoperiod. So they're measuring the amount of light that they have. And when the USDA conducted these types of experiments, they found that not only was this phytochrome protein responsible for flowering, it was also responsible for seed germination. So they were able to kind of look at the seed germination and the flowering to kind of figure out these types of things. And what they found was, is if they were just beaming it with some monochromatic light, in other words, one color of light. So we have red and we have far red. They just have, they're both kind of in the red color, but they have different wavelengths with them. And they noticed that one of the colors kind of induced the germination or got these plants to germinate, and the other one inhibited it. And what they were doing, if you look here, and I know some of these pictures are the exact same, but it's just kind of illustrating the point here. If you keep these in the dark, you can see that you're hardly getting any germination at all. And there are a couple of roots in there, and might be for a variety of reasons. They either were exposed to just enough light to make them germinate, or some sort of a mutation or something like that. But if you look down here, you can see when you beam it with the red light, you get a lot of germination. And then when you beam it with red and then far red, which turns it off, again, no germination, okay? And then here you get, they're playing with it now. Again, red, far red, red. Red turns it on, okay? You get germination. And then over here, red, far red, red, far red. Far red, of course, turns it off. And then, again, no germination. So that's basically what they were doing. And if you look at the flowering types of experiments, what they were able to show here was that you've got the plant making this, what's called Phytochrome red or PR, and the red light, this is the kind that optimally absorbs red light. The red light converts it into the PFR form. And this PFR form here is what's responsible for germination. And then very slowly in the dark, because there's no light really occurring, no red light, it's slowly getting converted back into this form over here, which was optimal for absorbing the red light and kicking this process back in this way. So this phytochrome switches back and forth between the two different forms of this phytochrome based on the type of light that it's beaming with. And you can see, of course, one causes the germination, one form of the seed, this form causes germination because the red light has converted it into that form, and this one shuts it off because the far red light has converted it back into the inactive form. So the active form of the phytochrome results in the transcription of these FT genes, which are just named flowering time. That's what the FT is. And this FT messenger RNA gets transported in the phloem. Okay, so there's the vascular system of the plants that we've talked about, to the apical meristem in the shoot, where it's translated into protein, and that protein enables these transcription factors to activate these flowering genes. Okay. So the big question is how do plants in nature kind of elicit a response to this light and begin germinating? So if the seeds get kept in the dark, they're making this PR form. And when the seeds are illuminated with sunlight, this PR form begins to get converted into the PFR. So in other words, the R and FR are just the kind of optimal wavelengths of light that these were found to be absorbing. And the appearance of the PFR is one of the ways that the plants are detecting this light. The adequate sunlight converts PR to PFR, and again, just like we spoke about a second ago, this is what triggers germination. So in the flowering response, what the scientists were able to do was show the effects of the red and the far red light on the flowering ability of the plants, because they figured it out with the germination, they were looking at the flowering, and again, they found the two things were canceling one another. One undid the other. So when we're looking at this flowering, like we just talked about here, again, we're looking at these short day plants, okay? And these are actually, again, long night plants. So when this photoperiod is really short, we have a lot of darkness, we get some flowering, okay? But if we flash it with a little bit of light here, they found that there was no flowering. And this was effectively the same as that. These two were essentially the exact same. And what they were able to show is the opposite again with the long day plants. These long day were actually short nights. So if you look here, you've got a short night. This, even though it's considered to be a short day and a long night, what they've done is effectively made two short nights. And what they were able to show here is that this was just like that, and they got flowering. Okay, so you can see the flowers forming right here. And again, this is a long night, and these do not respond that way, so then you get no flowering here. And what they were able to show over here was essentially the same thing. This pulse of light now, instead of being very vague in terms of the color, they were able to show the effects of the red, and then the far red, the red, and then the far red, in terms of how it was controlling these two things over here. Okay, so if you just kind of look at that, you can kind of see how these things were kind of managing the amount of flowering that you saw. So now as far as pollination has occurred, so once we get these plants to flower, they've got to be pollinated, and pollination is the first step in the events that lead to this fertilization. Okay, and it occurs when the pollen from the stamen of one plant lands on the stigma of another plant. Okay, and pollination occurs either through pollination by the wind, okay, the dust, pollen, and stuff blowing around that a lot of people are allergic to, or animals, birds, insects, things like that that are carrying the pollen from one plant to another when they're feeding and on the nectar and the fruits and things like that. So the pollinators, of course, are attracted to the scents given off by the plants, and as well as how they look to the insects or other animals that are kind of using this plant, this flower, as a food source. And when these pollinators are getting the nectar, of course, like we just said, they're picking up the pollen and they're transferring it from one plant to the next. And there's from a coevolutionary standpoint, there's insects that kind of co-evolve with certain plants. And you can see here, if you're looking at the long proboscis on this particular insect, it goes right down into this flower, so it's able to pollinate this flower and this flower only when it drinks the nectar. So these two, this flower got longer as this proboscis got longer, and vice versa. So as this thing gets the nectar and drinks, it's also transferring the pollen. And if something were to happen to this organism here, this moth, this flower would also suffer the same fate. And the same thing is true of these birds. I think this is maybe a honey creeper or something like that. This bird pollinates a plant with a flower that's shaped just like this. And again, they kind of co-evolve together. So ideally, plants don't self-fertilize, and this is a way that you get a good mix of genes so that the future generations of plant are hardy and healthy and not susceptible to disease. And there's certain mechanisms that plants use to prevent self-fertilization. One is a dioecious plant. These plants don't self-fertilize because they're one of two types. They either have stamens on them in their male plants, or they have carpels in their female plants. So obviously they can't self-fertilize. But other plants, like a corn plant here, this is monoecious. In other words, there's one type and it's got both male and female parts on it. And the thing is usually these organs are going to develop at different times. So the pollen is going to be ready at a different time than the female portion. So this hair right here on this corn, those are the female portions of the plant. And what those things, each of those go to a seed. And when a pollen grain falls on the end, pollen tube grows down, fertilizes that seed, and then the corn is going to develop into a cob of corn. So if you've ever husked like sweet corn or something like that, and you follow the hair down, you'll see that these little hairs here go to each of the kernels of corn that are on the cob. All right. Other mechanisms that prevent self-fertilization, you have the floral organs. They're arranged in certain ways so that these plants can't self-fertilize. In other words, like if you look here, you see these male portions, these stamen, are well below this carpel right here, or this stigma, the sticky portion of the plant, and that's what the pollen is going to land on. So when they're below it, they're much less likely to self-fertilize. And there's also self-incompatibility, and this is a way that the individual plant and that of the close relatives are rejecting each other's pollen to prevent this development of these plants and producing weak offspring. Okay. All right, so fertilization occurs after pollination, and in this process, this is where a pollen grain is going to land on the stigma of the plant, and the pollen tube is going to grow down the carpel. And once it gets there, you're going to have this pollen tube that's going to grow, and it's going to deliver the pollen to basically the egg, and then we're going to talk about some of the things that are going to happen there. And in angiosperms, again with flowering plants, they have this thing that's very unique called double fertilization. And so two of the sperm are going to be in the pollen grain, and they're going to travel down the pollen tube. So you can see right here where they're traveling down the carpel. That pollen tube is growing down, and eventually it's going to get to the egg, and the egg of course is down here. And once it gets there, those two sperm are essentially going to do, two really important things. And one of them is you're going to take this polar nucleus down here, which is diploid, and you're going to fertilize that. You're going to produce a triploid nucleus. So when you look here, this is the endosperm, or the interior kind of portion of the seed. That's going to be triploid. And then the zygote, the thing that's actually going to develop into a new plant once it germinates, this of course is going to be diploid right here. So what this is going to do is this is going to cause this endosperm right here to grow very rapidly and store a lot of food, so that when this seed germinates, it's got some energy to kind of use it before it gets above the ground, which it can then of course start performing photosynthesis and make its own sugars and nutrients that it needs. And this is one of the reasons why, of course, we make use of the seeds of these plants as food, because that's exactly what they are, is they're nutrients. Okay, and this is an avocado seed here, and then this interior portion right here, this is all the endosperm. It's all a triploid nucleus, and I don't know if people eat that or not. I don't, but this is the zygote down here, and this is what would develop into another avocado plant. And while that seed is buried underground, it needs, of course, some energy so that it can get its root and its shoot going and get that shoot above the ground and start performing photosynthesis. It's going to call upon all of these nutrient stores that we have here, as an energy source to get that shoot and, of course, the roots growing. And what double fertilization is doing is it's helping to prevent wasted resources. In other words, if the nucleus or the zygote, of the plant doesn't get fertilized, there's no reason for this plant to continue to grow, and there's no reason to develop this big, elaborate, nutrient-rich seed if there's no zygote in there. So double fertilization is really helping to prevent wasted resources. Okay. And then after double fertilization, after this fertilization has occurred, each of these ovules inside the carpel here, this is going to develop into, of course, a seed, like you see here with the peas. And if you're looking at this actual pea plant here, kind of see right in there where there's a pea. You can kind of see right in here where there's a pea, and so on and so forth. And each of these things, of course, are going to become this multinucleated supercell that's got lots of nutrients in it. And what it's going to do is it's going to nourish this growing zygote as this thing is starting to put out a root. And then once that root and that shoot get going, then, just like we said a few minutes ago, it can kind of take care of its own business. All right. And then if you're looking here, these fruits are going to arise from these flowers. So you can see we've got a flower right here. This was a raspberry flower, of course, and then this thing is going to develop into a fruit. Here's an apple blossom that's starting to swell after it's been pollinated and fertilized. And if you look down here, you can see these are all the stamens that had the pollen on it. And then this carpel, this thickened wall here, is what's going to kind of develop into this fruit. So you can kind of see how these plants are basically developing into a fruit containing one or more seeds from the actual ovary of the plant. Okay, so when these seeds are developing, the walls of the ovary are actually what you're eating when you're eating fruit. The thickened walls of the ovary are actually developing into this fruit. That's what this pericarp is. So when you're looking here, you can see here's the flower of the plant, okay, in this pea example right here. And then you can see here, this is the ovule. Okay, and then you've got the ovary kind of surrounding all of these eggs right here. And then what you have also is this stigma on the end of this thing. And this thing is actually what is, was formerly the flower. That's what's going to develop into the part that we're going to eat, or the part that's going to give rise to more pea plants. Same thing with this raspberry plant here. Okay, you've got numerous carpels here, and all of these things are going to kind of fuse together, and you can kind of see the stigma and the ovaries and sort of that sort of thing on these plants here. And then the same thing here with this multiple fruit, and we'll talk a little bit more about this as well here with inflorescence. And what this flower is doing here is these things are all starting to grow together to give rise to this pineapple. So simple fruits, you have a single carpel, just like with the pea plant here, like we just saw, and you've got peas and peaches and certain tree nuts that are growing this way. And again, you can see here these are the sepals, these modified kind of petals right here, and then inside of this is where the flower actually contained the female portions of the plant, as well as, in this case, you can see the stamen here, these male portions of the plant, and those what gave rise to the pollen. And this was that sticky stigma right up here. So the pollen landed on this, the pollen tubes grew down and fertilized all of these seeds that were in there and so forth, and then that's what's going to develop into these peas. Okay? So now these aggregate fruits, these things are like raspberries, and you can see the carpels. If you ever look at the flower of a raspberry or strawberry or something like that, you can see these carpels. These things are each, all of these things are going to get fertilized right here, and you're going to get pollen tubes that are going to grow down, and then in each one of these little kind of fruitlets that are on there, of course, there's a seed. Like raspberries have lots of seeds, and blackberries and things like that. And so this flower is what's actually going to develop into the fruit. And you can see again the thickened walls of these ovaries here are becoming the fruit, and they're housing the seed. And if you take a picture of an actual raspberry, you can see all of these stigmas that are on there that were once fertilized, And then these, of course, right here, these are the ovaries that contain the seeds, and that's the fruit. Okay, and then of course they taste good. Animals eat them, scatter the seeds with a pile of fertilizer, and then a new plant grows. All right. And then inflorescence, this is like a cluster of flowers. And when you're looking at this cluster of flowers, when these things, each of the flowers get fertilized, they start growing together. And then so what you get here is a multiple fruit. In the case of a pineapple, all of these flower pieces kind of grew together. And of course, there's this kind of central core running down the middle of it that they're all growing out of. And that's what a multiple fruit is. And that's from inflorescence. Okay. So, fruits and seeds are usually kind of ripening and when they're all completing their development. So like a peach, when the peach pit basically is where the seed is, when that thing is ready to be kind of planted and grow into a new tree or whatever, that's about the time that the fruit is ripening. Same thing with the soybeans. These things age, they dry out, and these seeds now are ready to be planted, and then they will soon become a new soybean plant. Okay, so now with seed maturation and development, as this seed starts to mature, what it's doing is it's preparing for dormancy. It's drying out, like we just said. But once it breaks dormancy, in order to do so, it's got to find an optimal condition, and that's where it's going to be, warm soil. It's going to have plenty of moisture, things like that. Imbibition, IMBIB. I'll spell it out here. Imbibition, that's where the plants are going to absorb water, and when they absorb this water, that's what's going to also help them break dormancy. So if the soil conditions are right, it's warm enough, the enzyme systems are going to kick in, and the plant's going to grow a root and a shoot, and then it's going to start to grow and develop into a new plant using those nutrient stores in that seed. Okay, now as far as seed production goes, plants often produce tons and tons of seeds because the chance that any one seed is going to survive is very low. So just like with asexually reproducing organisms, there's lots and lots of organisms produced because many of them often die. The same thing is true with these seeds. And if you think about plant reproduction, plants use both sexual and asexual reproduction, just like many animals do. And there's some advantages and disadvantages, and you should know a couple of each. For instance, one of the advantages of asexual reproduction is you get these offsprings that sometimes are more hardy than the seedlings. In other words, they're able to quickly form a plant. But one of the disadvantages is they're susceptible to the same disease. With sexual reproduction, of course, you get lots of genetic variability. Due to the changing environments, they have the ability to adapt. And one of the disadvantages, of course, is you don't always have a mate, have access to the things that you need in order to give rise to a new individual. So if you can go through and think of a few more advantages and disadvantages, it'd probably be a good idea. Now, as far as seed dispersal goes, once the seeds have been produced, there's a wide variety of mechanisms that they are, have kind of evolved along the way, so to speak, and allows them to be dispersed. So some seeds are buried within kind of barbs on these plants that stick to animal fur and so forth. Get transported around and fall off, and that's a way that they can kind of distribute themselves. Other fruits and things taste good, so animals eat them and carry them around. Some are dispersed by the wind and so forth. So hopefully this helped kind of go through some of the things with you. And of course, if you have questions, make sure that you write those down, and then we'll be sure to discuss these in class. All right, so we'll see you soon.